Hi, good morning, or probably midday-ish for you. But let me just jump straight into it. So this is a live streaming webinar from Zimmer and Peacock about biosensor development, manufacturing, and troubleshooting. Just to say that I do have technology in front of me. So like many ZP webinars, this won't just be talk. We'll also be doing some live demonstrations as well. So just FYI, everything's going live, and there will be some demonstrations as well. With that said, let me go forward. Now, it's probably worth saying there's a few people attending today that I've known for well over a decade, and these people are, as experienced, if not more so than even myself. So I really appreciate you coming along this afternoon with FDA-approved products on the market. So thanks so much for coming. With that, let me shoot forward a little bit. So it's biosensor development, manufacturing, and troubleshooting. It won't all be talk, and it also won't be the kind of material that you can just find in a textbook. I will talk about the steps in research. I'm going to kind of talk about, if I was developing a biosensor, there's certain characteristics that I'm looking for. I mean, they're obvious, some of them, but the characteristics are obviously, can I detect the analyte? Can I detect the analyte action in a clinically useful range? What's my selectivity / specificity like? And really, can I get reproducibility in manufacturing? What's the biggest problem in commercializing biosensors? Well, one of them is raising money. Without that, you can't really achieve anything. But one of the technical challenges is actually reproducibility of manufacturing, and I will touch upon that this afternoon. It wasn't as advertised, but I will be doing a bit of a teardown on the Abbott Freestyle Libre 2. I think a number of you will have an interest in CGM, continuous glucose monitoring. And I think there's some lessons. If we're going to have a webinar about development, manufacturing, and troubleshooting, then I think looking at what successful products that are being manufactured at scale, how they do it, I think is a valuable lesson. So we'll have a look at the Abbott Freestyle Libre 2. And I'll also have a bit of a teardown on a glucose strip as well. Because I think it's just interesting, actually, that even though these two, when I say these two products, a glucose strip and Abbott Freestyle Libre 2, they look very different. But in fact, when you really peel it back, you actually find out that they're actually pretty similar products. I'll also talk about the processes that go on to make a glucose strip. Not because I want anyone here today to make a glucose strip. I only use it to illustrate that I think the glucose strip is the blueprint on how to actually make a low-cost, scalable / manufacturable biosensor. And people keep trying to reinvent manufacturing processes and technologies. It's like, if you want to make a biosensor, I'm super biased. Electrochemistry is a good science. And if you want to know how to manufacture it, just peel back the layers of a glucose strip. Anyway, I'll cover that today as well. I'll do a quick demonstration. As I say, this won't all be talk. I do have some hardware in front of me, and I'll be illustrating some of my points about reproducibility by doing a demonstration with some printed electrodes shortly. So there's my phone. If you can see my camera, I've got some sensors here, and I'll be doing a demonstration as well. So as I say, we're not just going to talk, and I'll try to be efficient with time as well. I will talk about manufacturing reproducibility, and I'll also talk about the cost to get to market. As I say, I can see a couple of people here who've just as much experience as I when it comes to the cost of going to market. Just watch out. Yeah, we will discuss that, and some of you may get sticker shock that it's more expensive than people actually realize. So the reason that we're here is because I think really point of care diagnostics in some ways started with Johnson & Johnson at the beginning in the 80s doing glucose. They were doing a glucose meter for diabetics. In those days, they were actually doing it optically. But then at the beginning of the 90s, you had companies like MediSense and slightly later on Inverness Medical making glucose strips. So we got these self-monitoring blood glucose strips where, the finger is pricked. The sensors themselves are quite low cost. The meter is, small form factor. And the glucose strip serving diabetics has really kind of proven a value. And it's a good platform with which we can copy when doing other biosensors. So when I hold up a kind of example of the latest kind of readers from Zimmer and Peacock, though this is bulkier than a normal glucose strip, you'll see a lot of design principles that have actually come from the glucose strip world. As just a side note, I started my career making glucose strips in 2000. I'll also talk about CGM, something I'm really in. When I say really into, I mean I've got a lot of industrial experience in making continuous glucose monitoring, not on the Abbott Freestyle Libre 2, but I did live in California for a good 8 years and got very involved in CGM as well. So I'll be talking about manufacturing of these as well. When it comes to the research part, now I'm always coming from a perspective of electrochemical biosensors. I mean, the most obvious question when making a new electrochemical assay, when I talk about electrochemical assays, I could be talking about proteins, I could be talking about cytokines, I could be talking about small molecules like lactate, glucose, ions, sodium, potassium, etc. Does the sensor respond to the analyte? I know it's an obvious question, but that's tick box number one, and it's almost the easiest one to do. That review the literature, you can always find some formulation, especially in the electrochemistry space, for detecting particular analytes. So yes is quite easy to achieve. So when thinking about the research path, does my sensor that I'm developing, and hopefully you're developing it on ZP products, quick product placement plug from me. But if not, you can still be successful. But so hopefully you are detecting the analyte. If not, go back, review the literature. That especially in the domain of electrochemistry, most molecules that you can think of, if you Google them and put electroanalytical in front of it, you will find a paper. Sidebar note here, but there's about 25,000 papers produced per annum, or probably published per annum, in the space of electrochemistry. So it's a rich gold mine to mine when looking for principles with which to detect analytes. The next question is it operational, that sensor, in the clinical range? So I'll give you a really obvious answer I'm seeing a lot of at the moment. So there's a group of molecules called PFAS. There's that Mark Ruffalo. There's a film called Black Water, and it really talks about the sort of Dow chemicals, and PFAS was this group of molecules that were highly fluorinated alkyl substances, and they're causing toxicity. Now, the EPA regulations on that says that these molecules shouldn't be above 4 ppt. That's not parts per thousand, that's parts per trillion. Now, the reason I bring it up is because then when you read the literature, people are doing detection of these molecules in parts per million. So they're publishing papers saying I've got a sensor specific for PFAS, but in reality, they're 3 orders of magnitude higher in concentration when publishing as to what they actually need in the application. Now, I know that's not a medical not a medical example, but it is a current example of what's going on in the literature. So easiest thing, one of the easiest things, is actually just to be able to detect. Then can you detect it in a clinical range? And then it comes down to really what's your specificity, what's your selectivity. So antibodies can often be quite specific, whereas maybe an enzyme has some cross reactivity. So glucose oxidase, maybe it reacts a bit with maltose, maybe it reacts a bit with fructose. So then we would sort of describe its selectivity. Whereas if something just only binds to one protein, let's say it's interleukin 6, and you've got the antibody to interleukin 6, then you may actually have a high degree of specificity there. So specificity is quite achievable. Groups of molecules like antibodies could be specific. Enzymes can be quite specific as well. I can give you an example of where selectivity. There's an ionophore for detection of sodium, and it has some cross reactivity with potassium and probably ammonium. So that's one where you've got a bit of a problem with selectivity, but hopefully you're selective. And if you're not sure where to start with that question of selectivity, you can have a look at ISO 15197, which is the ISO standard for glucose strips, because there it'll talk about all the common interferences that a glucose strip has to be tested against and at what concentrations. So that's a good place. But of course, I also appreciate with this audience, I think a lot of you know it, so I won't bear a long time on that. But if you don't, and you're wondering about, well, if I need to test on my selectivity, my specificity, and my resistance to interference molecules, where should I start? ISO 15197 is a good place to actually start with that list. Ooh, the elephant in the corner. is my sensor fabrication reproducible? So the elephant in the corner is it's very hard to actually get reproducibility, probably very hard to get reproducibility in biosensors and hard to get a high yield in biosensors. What I mean by high yield is if I make 100,000 sensors, will I get 100,000 good sensors or will I get 90,000 good sensors? So reproducibility and yield are probably the two toughest things to achieve. If you can achieve, let's say, reproducibility of a relative standard deviation of less than 20%, then you're probably in a good place. But especially in an academic setting, and I know not everyone here, is in an academic setting, but in academic settings, people are always trying to work on reproducibility, and you're not going to achieve it. The balances are not calibrated, routinely. The pH meters are, are off slightly because of what somebody previously did. The environment is not controlled. So in a industrial setting, yeah, you can get reproducibility. You can have a clean room. You can have everyone gowned up. You can control the environment. You can control temperature, humidity. You can control the incoming materials. You can have process controls. You can get reproducibility. But if you do have a sort of academic or slightly academic background, honestly, don't think that academia is ever going to really achieve reproducibility. Now, it's a bit of an opinion, but who can name that biosensor that came from an academic lab, and when it came out of that academic lab, it had great reproducibility? Certainly not at scale. So you can achieve in the research phases response to Analyte. You should be, well, if you should in the research phase get to the in the clinical range. In the research phase, you should be able to test your specificity / selectivity and test it against interferences. But in that research phase, you may not hit reproducibility, and that's because that's really a development phase. I would argue with that. Now, the progression along here, I sort of say, as we progress from Analyte detection to clinical range to specificity to reproducibility, that progression becomes difficult. And I just actually sort of jokingly put this as a log scale here, because I actually think that reproducibility, etc., is really an order of magnitude harder to achieve than the initial proof of principle when you first detected that Analyte. Most academic papers are really getting to technology readiness level number 3. They're really just not just, but they are proof of principles. And that can be achieved, in some months with a good scientist. But reproducibility can take some years and a lot of investment. Now, what can we learn? So something that I think we don't do enough of this. And I have a couple of my colleagues who are here this afternoon. You do have this, you do have this experience of what have people done in the past. But I think it's a good place to sometimes start. So if we do a partial teardown on a CGM, so for some, I'm sure most of you know, but a CGM, a continuous glucose monitor. This is the Abbott Freestyle Libre 2. It's those sensors that you see people walking around, and because a lot of you are from North America, Dexcom is just as familiar with you there as well. If we do a teardown on this, it's kind of easy enough to remove this white, I don't know if I want to call it a cheese slice, and at which point then you're kind of looking at this kind of PCB. So I will be going through this anatomy quite a bit, but I'm an electrochemist, so I immediately start understanding this because I can sort of see, there's 3 contact pads there that will become significant to me in a minute. So those, when I've removed that sensor, in fact, I've got it here. I'm not going to focus so much on the electronics on this, but just FYI, the electronics on the Abbott Freestyle are actually using an MSP430 chip. Some of you will know that Texas Instruments make the MSP430 chip. It's the chip that's in a lot of glucose meters. I'm not recommending the MSP430 chip. I think the MSP430 chip's been around for like 25 years, which is a good thing because it's probably pretty robust at this point. The supply chain is very robust, but there are some, let's say, more modern semiconductor chips these days for doing electrochemical analysis. Anyway, I did pull apart a Abbott CGM. I don't work for Abbott. I'm not any affiliation with them. I just pulled it apart and had a quick look at it. And what we find pointing back at us is actually 3 contact pads, which is music to my ears because I'm an electrochemist. So if you ever hang out with electrochemists, you'll hear us talk about We like our working electrode, our reference electrode, and our counter electrode. So the Abbott glucose sensor is almost like a classical electrochemical cell. It's got a working electrode, a reference electrode, and a counter electrode, which makes a lot of sense because it actually came from a very classical electrochemist, a guy called Adam Heller, who is in Texas. He's a professor there. He and his son founded a company called TheraSense. And Adam would, if you read, not that I know Adam, but Professor Heller, if you read his papers, then you will sort of see very classical electrochemistry or working electrode, counter electrode, reference electrode, and lo and behold, it makes it into the actual product itself. Now, if we zoom in a little bit more on the sensors, when I was doing this, I was expecting something else, but what I saw, and some of you will sort of say, well, we knew this anyway, but I'll say it for the other people in the room. When we zoom in on this, we find ourselves looking at a little sort of rectangle. So at least this part here makes me think that this is the working electrode. Now consider the scale on this filament that Abbott likes to call it, is on the order of a hair's thickness. So it's a pretty tiny little object, and somewhere on there's this little yellow square. So that's probably the enzyme layer. It looks like a sort of looks like glucose oxidase. Glucose oxidase is often yellow, so I suspect it is glucose oxidase. Now, what I found interesting about this analysis was I was expecting this object to be cylindrical, and I'm sure many of you actually know that it's not cylindrical. It's actually more sort of rectangular, like this. We can learn a lesson from this, to be frank with you, because when you have a cylindrical object, and there's another CGM on the market which is actually quite cylindrical. So when it's cylindrical, that forces them to manufacture in a certain way. But because Abbott is actually manufacturing on a rectangle, and I'll describe, or flat plane, I'll get into that shortly. They're avoiding the problem I'm about to tell you. So at Zimmer and Peacock, we do a lot of work with wire sensors. These are cylindrical. We do a lot of work with microneedles these days. Sidebar comment, the company that's kind of leading microneedles at the moment is a company called BioLink down in San Diego. They've raised about 340 million in the last 4 years. It sounds like they are having success with the FDA, and they're using microneedles. Quick plug, Zimmer and Peacock also has microneedle technology. We have a patent portfolio of 50 patents around that, and we do contract development, contract manufacturing, specifically on microneedles. Anyway, off that point, let's talk about this. Why is it an advantage for Abbott not to be cylindrical? And this quick answer is this, that if you're trying to do drop casting, or dropping nanoliters or picoliters onto a cylindrical surface or a curved surface like this, then gravity is not your friend. Things will sort of flow away from you before you know it. I know it's obvious, but having tried to pipette on top of objects when they're curved, then the glucose oxidase, for example, will just get away from you very quickly. But Abbott have been clever, and I got a suspicion that Medtronic also do this as well, because I'm pretty sure that Medtronic, when they were manufacturing their CGMs, were on quite a planar substrate as well. That they're on a planar substrate, so when you do drop casting, for example, it may spread, but it's not going to run around the curved surface and onto the floor of the chamber. So Abbott are giving themselves a manufacturing advantage by manufacturing in a sort of planar substrate. I know it's obvious saying it, but I didn't actually realize until I'd taken these images that that's what they were doing. And this means that what Abbott is doing is actually probably manufacturing electrodes more in a kind of, I'm just going to grab a sheet. They're manufacturing on a much smaller scale, but they're probably manufacturing like this, where they actually have sheets of their electrodes, much smaller. But then they're functionalizing by doing deposition, probably from an XYZ robot. Now, I say probably because I don't have the first-hand knowledge. I'm just doing reverse engineering by looking at it and using some experience. So they're probably manufacturing in a planar way, and then they're drop casting on their hydrogels in that particular case. And I sort of zoomed in on that. Now that looks very dissimilar from a glucose strip. I've got a glucose strip here. But actually, I think it physically looks dissimilar, but Abbott obviously have a lot of experience in glucose strip manufacturing. So I suspect that they've actually learned something from the glucose and they actually apply it to the CGM. I'm just going to do a quick jump down or jump into a partial teardown of an SMBG, a self-monitoring blood glucose strip. Why? Because everyone in the world of diagnostics all say, I want to make a low cost, easy to use, point of care diagnostic. That's a glucose strip. Everyone says it, and then they go off and do something which is kind of microfluidic and valved and reagents and calibration. It's like, no, 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 no. If you want it to be low cost, easy to use, manufacturing scalable, then you should really be looking at a glucose strip and having a workflow that's like a glucose strip as well. Because everyone uses the term low cost, easy to use, and then they make something that's really lab on a chip. And I think lab on a chip is a design philosophy that leads people to complexity. Anyway, let's do a teardown on an SMBG strip. So now the SMBG strip, unlike a CGM, a continuous glucose monitoring sensor, it has to serve a couple of functions. It has to support the enzymes so that the reaction can take place, but it also has to actually bring the sample to the sensor. So there's a bit more complexity in a glucose strip, but if we tear one down, we can essentially sort of reconstruct it when we want to make a new sensor, sort of like a sepsis sensor or, something like that. Okay, so what's different about the blood glucose strip? It has to suck up the blood. So we will describe that manufacturing today as well. So I pull this apart, and I know many of you got this kind of experience anyway, but the first thing we find is, there's actually a top cover and then there's a base layer. Now the top cover, if we kind of zoom in a little bit on that, this is the kind of where you put all your logo and stuff, but the real work end is actually down here when we've got this kind of notch cut out. So that notch is really describing the walls of a capillary, and then there's a transparent cover over the top of it, which is actually it's actually forming the wall of the microfluidic chamber. When I first microfluidics sort of started appearing at least in my vocabulary, around about 2005, and when I looked at it, I thought, this is the glucose strip, isn't it? There's always been microfluidics in diagnostics. It's called the glucose strip. Anyway, it has a microfluidic chamber. Just FYI, most glucose sensors today are actually operating on 300 nanoliters of blood. So if anyone's ever asking themselves the question, how much sample do I need in order to do an analysis in a drop of blood? If it's something like glucose, which is at quite a high concentration, 5 millimolar, then 300 nanoliters is all you need. And that's what that chamber volume actually is. If we look at the base layer itself, this looks like this particular one. You can screen print, you can do chemical vapor deposition. This one looks like it may be chemical vapor deposition, and they've used a kind of light or a laser to scribe particular conducting tracks, and then there's a black dot which is probably contains all the reagents and all the enzymes. So they look different. I mean, this Abbott Freestyle Libre 2 looks very different from our glucose strip, but actually they're really more similar than I first would have realized, which kind of says this, troubleshooting, what lessons can we learn? Don't reinvent manufacturing. What I mean by that is, when you're doing your development, yeah, sure, develop a new assay, develop a new aptamer that gives you specificity, develop a new way of getting the most signal out of that assay using some new electrochemical method that you want. But please don't reinvent the manufacturing, because if there is a manufacturing route. There are vendors out there that will give you printing, deposition, cutting, laminating. All these processes exist, and they're known to work and they're known to scale because it's called a glucose strip. And lo and behold, they may be even using that technology on these CGMs, continuous glucose monitoring. So be really, really careful about reinventing manufacturing. I mean, if there's nothing else I can say today, then I would like just that message alone is what I'd want to really say about this. I did mention it earlier on, but the glucose strip has an additional functionality. It's not just a substrate on which we're depositing the enzymes. or the ionophores or the antibodies or the aptamers. The glucose strip's got a bit more that it actually is creating the capillary. So we get something called capillary fill, which if you're going to make a medical diagnostic, the way the sample comes onto the sensor can really be part of the signal. So you really need to be able to control that process. And the way that the capillary system works is a really elegant way of doing it. Ironically, it didn't come from academia. I think it came from either, I think I want to say Unipath. There's a company in Bedfordshire in the UK that originally held the patents on capillary fill. Anyway, capillary fill, great technology, off patent these days. And if you're trying to make an easy to use, low cost sensor, don't use lab on a chip principles, just use capillary fill principles from the glucose strip. So at Zimmer and Peacock, we do work on capillary fill quite a bit. I mean, if I was to do a cross section on this ZP product here, what we're going to do then is we're going to understand that the enzyme is at the top. We've got our spacer layers that create our microfluidic chamber and our base layer. So if I draw a line across here, I mean, we actually use a lot of thick film printing. I'm going to come to thick film printing again in a bit when I talk about reproducibility, but we have a substrate and we print on top of it. We put an insulator, or some of you will refer to it as a dielectric, and that's kind of what's happening across that cross section. It gets a bit more busy at the top end of this, where we're actually doing the sensing. And we have our sheets, we have our prints, but in the case of a glucose strip, we've put an enzyme down there. And then we've actually put this sort of laminate on top of that to create these walls. And on top of that, we put a clear cover top layer to create the capillary chamber. So I was referring to the capillary chamber earlier on a glucose strip, and that's like 300 nanoliters. The chamber on this one is bigger, it's about 3 microliters, but 3 microliters is still smaller than a sort of drop of blood. So lessons to be learned from here is the glucose strip has to serve a second function that the CGM doesn't have to serve. And the second function is it controls the volume of the sample, great. And it also controls the filling of the sensing chamber as well. And it's definitely scalable. You know, if you ask, and can be low cost. Now, don't forget, glucose strips have had a good 40 years worth of R&D gone into them. You know, but what does it cost to make a glucose strip at scale? 2 to 5 cents. What can they sell for? 50 cents. So it's still, even though it's a commoditized product, it can teach us a lot about making manufacturing low-cost sensors. So when you do look at the workflow for these things, you know, a glucose strip, you know, consists of at least 4 parts. You know, we've got the enzyme solution itself, we've got the base layer, we've got the spacer layer, we've got the top layer. So the base layer is kind of, I've put it here as often PET, polyethylene tetrathalate, onto which you will maybe do vapor deposition or you may print, but we'll, at ZP, we'll print. And then we'll also want to isolate or insulate the conductive tracts, because we only want certain parts of the conductive tracts to be exposed. Otherwise, we have an uncontrolled working electrode area, and that will really mess up our reproducibility. But at some point, that formulation, it could be a molecular formulation, it could be an aptamer formulation, it could be an antibody formulation, it could be an enzyme formulation, it could be an ionophore formulation, that has to come down onto the sensor. And I was talking to a company today that does kind of picoliter deposition. So you can, there's all sorts of products on the market, but we do use an sort of XYZ piezo device print head for depositing liquids. At which point now we've got our enzyme on top of the sensor. So that kind of product at this stage may look like something like this, but you know, doesn't have the microfluidics on top of it. So now we want to start putting the microfluidics on top of it. So we may use a double-sided adhesive material to laminate and create the walls of our chamber. So we'll then have to sort of laminate the double-sided adhesive on top of our sensor. And then there's a moment now where we want to bring the top cover over the top of it. We often use a transparent top cover because it really gives us visual feedback that the design of capillary fill that we've designed is actually working, because we can generally see the blood flowing into there. Now, the reason I bring all this up is because there's at least 15 processes going on here, all of which will be controlled. So you cannot really manufacture a biosensor outside of a contract manufacturing organization, or you've established the whole production line yourself. But not everyone here today is an academic, but to the academics in the room, you do have to form some sort of company really to put together this kind of device, at least put together this kind of device in a repeatable manner. At which point now you're starting to get sheets of these sensors. So then you have to cut them into strips, then you have to singulate them, and then you can finally get around to packaging. So there's a lot of words there, isn't there? But it's not that straightforward to even manufacture something that seems as relatively simple as a glucose strip. Now I've put here a quick demo to remind myself to do a quick demonstration. So forgive me for a second while I stop sharing that screen. I'm just going to share the desk in front of me. So at Zimmer and Peacock, there's something we really have to learn from the tech industry. The tech industry will talk about MVP, minimally viable prototype. This is starting to find its way into point of need, point of care diagnostic world. But I've seen so many companies spend 15 years developing products, and at the end of it, I've got a specific company near me in Cambridge that did this, and after 15 years, nobody wants the product, and it's just a complete waste of time. So we need to get to productization much quicker than historically has been the case. So at Zimmer and Peacock, we actually have an instrument like this. Now, at the moment, my phone says lead and copper because I've been using it this afternoon to detect lead and copper. So I understand, oh, that's interesting, lead and copper. This is a webinar that's primarily about biosensing and probably primarily about medical biosensing. But just let me take that sensor out and let me put a new slightly different formulation ZP sensor into here. So I'll drop that in. So just note the handleability of this, the way that, we've obviously designed it to kind of be, my finger doesn't go onto the sensing area. I can put that in. There's no slots. There's been a real trend to making point of care diagnostics all have slots in them, and slots are quite awful actually. Have you ever tried? Well, I'm sure you have, but slots can, things bend, things break, the connections quickly break. But we've got quite an elegant way of getting the sensor in and out. Anyway, that said, you're not here to see me test the quality of drinking water. You're here to see me possibly talk about medical diagnostics. But what I'm going to do is I'm actually going to take my phone. So I'll come back for a second. Just go into here, and I'm going to just scan that. So now my phone says caffeine sensing. Just want to give you a sense that I can go between analytes really quite easy. So let me just go back to here again. I'll just scan this QR code. So now my phone says lead and copper. So what I'm demonstrating to you is I'm able to go between different tests. So when people come to us and say, oh, I want to do a rapid, development of something that's quite new, we're able to actually just take this piece of hardware, which we call the sensor, or we're able to develop an assay on one of our kind of screen printed electrodes and do an app really quickly, and actually that meter is good to go. So what I'm going to now do is I'm just going to give me a second. It's not because it's 7:30 here that I'm drinking Red Bull. It's just I want to demonstrate some of our technology really quickly. So let me grab that Red Bull sample, which I am doing. So you could sort of consider this like a urine sample. I mean, funnily enough, it's slightly yellow as well. So, but you've come to a ZP and you want to come up with a new test. It's actually for urine. So because it was for urine, I've sort of advised you that actually we're going to have to do a compromise here, because in fact urine can be quite variable. So in this particular case, I'm just going to add a drop of buffer. So I've done a 1-to-1 addition of Red Bull and a buffer. In fact, I added the wrong buffer. I got two buffers in front of me, so I actually added the wrong buffer. So I'll just do that one more time. But I'll do it one more time and nice and quick so I don't take up your time. So I will go in, grab my Red Bull, which suspiciously looks like urine, but it's not urine, it is actually Red Bull. I'm going to grab my buffer, which I will do, so I can do this sample prep. And so if I was thinking about grabbing some early clinical work, or proof of principles, this is probably the workflow that I would actually suggest. So it's a bit manual because I just did that step. But that said, let me just put that drop onto the sensor like that. So I've just added 50 microliters onto the sensor. Electrochemistry is great because it really doesn't require that much sample. I mean, that was 50 microliters. Now, what I'm going to do now here is I'm just going to name this sample. So I'm just going to name this sample Alistair, because then I can find it later on. So I'll do that. I'm going to hit continue. One of the design principles that we do at ZP, and electrochemistry really allows this, is actually we like to make fast assays. So this assay is running, it's going to take about 20 seconds. I started my career making glucose strips. So those run in about 5 seconds. So I'm getting the raw signal, but just watch the next stage now. So I've just run Red Bull, and it's given me the answer and said to me this, right? It says milligrams per gram, 0.29. Now, what I've done is I've taken Red Bull, and you can check this out on ChatGPT. Ask ChatGPT what the caffeine concentration of Red Bull is, and it'll tell you it's 0.3 milligrams per gram. So understand that I've just measured the caffeine, and I'll show you the data on this. And now it's got 0.29. So I have around 0.29 up. That's 0.3 milligrams per gram. Now, the reason I've done all of that is because it really just comes down to something that I want to really emphasize: reproducibility. The only reason that ZP can rapidly develop assays and put new products out there at quite a rate is because we're controlling the reproducibility of these guys. That's really the secret. And so there's so many materials entering the R&D market, especially in the screen printed electrode area. They're not reproducible. So people are really being held up just because of that reproducibility. Now I want to just, I'm going to share my screen with you for a second because my phone says upload complete. So what I'm going to do is I'm just going to do a quick refresh on this. Hopefully it hasn't logged me out. Right, so I can see a data here. It's called Alistair. So that's what I named my Red Bull sample. If I go up here now, you'll be able to see it. So I can see my signal. It's Red Bull. I can see a bump here. My software has actually picked up the bump. It's measured the height, and it's converted the height into a concentration. Because nobody really likes electrochemistry, especially doctors and nurses. So we have to very quickly be able to convert the raw signal into concentration. And that concentration actually went back to my phone then, and that's what you saw on my phone. I got a few more slides on this just to kind of illustrate it a little bit. But even though that seemed like a very strange demo to talk about caffeine, what I was really demonstrating was the reproducibility of that. So what you saw going on there was I used the QR code to change the platform from being it's a potentiostat to actually being a Analyte specific system. Took a sensor, put a drop of Red Bull on it, used the QR code. The QR code told the app what to do. The app told the hardware what to do. The hardware got the data back from the app. It sent the data to a cloud system called Julie. So we actually crunched that data in real time. So imagine that you're actually doing clinical work or you're developing a new technology. The first thing that you get is you get a ring from a doctor saying it's not working. We're getting ridiculous results. Well, of course, with this cloud system, we can actually log in and see the data in real time. And we don't need patient data in that. So he doesn't have to have that whole HIPAA compliance part. But anyway, the data goes up there. We process the data. And lo and behold, I got 0.29 and my slides say 0.3. So I knew I was going to get the right answer. Why? Because the biggest problem in biosensor development is actually manufacturing repeatability, and that's something that we've really worked upon. So I knew I'd get the right answer because we've put a good decade's worth of effort into manufacturing. The fact that we got repeatability in manufacturing means that we had a simple assay, it was an accurate assay, and there was no calibration. So many actual devices, especially in the point-of-care diagnostic space, are really using this design principle lab on a chip. They've got valves, they've got fluids on board, they've got wash steps on board. They're really just taking the lab assay it is and just applying it, and it's not going to be simple at all. The reason we can get away with simplicity, accuracy, and no calibration is really the reproducibility of the manufacturing. And if you haven't got that, then it's very hard to get low-cost, scalable products. If you can't get repeatability, I tell this to my academic colleagues more than anything, because in fact your electrodes are not reproducible. And that's a plug because we would like people to use our screen printed electrodes. But the reason I'm able to do this is because, in fact, we do so much testing of our screen printed electrodes. We really, every time we manufacture a batch, we're testing them at scale, making sure that the batch is within, the mean, the standard deviation, the relative standard deviation, the median, the min, the max values. We're doing proper process characterization through the product to make sure that what we're using in our products is good, and therefore what we ship so people can use it in their products is also good as well. So there was a bit of the demo, and then but the demo was successful because of the reproducibility of our screen printed electrode. Final bit, so final stretch here, just to kind of say that I think some of you have actually got direct experience on these products, but let's just talk about cost for a second. I mean, the iStat came out in about 1983 and cost them about 50 million in 1983 to get to market. Abaxis, which is a company up in Union City, for example, now owned by Zoetis. I think they may have been bought by Zoetis for about $1 billion, but they came to market at the end of the 80s, and their cost to get to regulatory approval was about 40 million. So I've kind of done a little bit of a survey on all these different companies. Epocal, pretty similar to iStat because it's the same founder. In 2000, 40 million. Sphere Medical spent at least 30 million, never got to market. Tearlab, it's a teardrop osmolarity test. They were an outlier because they were only 7 million, but it's a very, very simple test. And then a few others. Now, the average for these companies to get to regulatory approval was about 38 million. If you take out Tearlab, it's actually more like 42 million. But I appreciate some of those numbers are quite old, like 1983 to 2005. So I did this exercise in 2021. If I did it today, we've actually had a bit of a hike in inflation. So in fact, but if I changed all those numbers, or rather artificially inflated them with the consumer price index, I can pretend that in fact all this work was done in 2021. And what this means is that I would estimate, and some of you will laugh and say, Martin, you're far too low. But on average, if you're going to do a point of care diagnostic, then you may be looking at something like 74.7 million. And if we take out something like Tearlab, that number can end up going to more like 83 million. So it would be quite, if somebody's asked me, what's the cost to get to FDA approval with a medical diagnostic, I can talk about 510Ks and different routes, but realistically, for like a molecular assay, something like sepsis or something like that, you're definitely looking at north of 100 million US dollars. I know to an audience like this, there's no surprises there, but it's amazing so many people who actually think that they're going to get there, in the low millions. No is the answer. Now, the timeline for this, I mean, the iStat took them 9 years to get to market. Now, it was the same founder who actually then did Epocal. And when he did Epocal, it took them 6 years to get to market. So the experience meant that they actually shaved 3 years off it. But, these are definitely the kind of lengths of time it takes to really get your manufacturing set up, get your enough products together that you can do your clinical trials, etc. So it's not necessarily a short sprint. And some companies, I've got Sphere down here at 9 years, but I think they may have even been 15 years. So it's no short sprint, and certainly it's not a cheap, low cost exercise. Right. So we're at 9:44. I've got 1 minute to go. So I will, and by the way, this has all been recorded. Give me a couple of hours and I'll send you out a recording anyway. So, academic papers are really getting to technology readiness level number 3. So they can often show the analyte detection, maybe do it in the clinical range, but they don't dig too much into specificity, and they certainly don't dig too much into repeatability. If we tear down a CGM sensor, then we actually find out that, they're actually manufacturing it in a manner that doesn't look so dissimilar to a SMBG, a self-monitoring blood glucose strip. The SMBG is the blueprint for making low cost, easy to use, manufacturable biosensors. Lab on a chip is a dangerous concept because it suggests that, if you want to have low cost, easy to use, and low cost instruments as well, then lab on a chip is not that. So if you can make your assay as simple as possible and borrow as much as you can from the glucose strip world. That would be, in my opinion, a really good strategy. Now, the only reason I showed you the caffeine sensors is because I wanted to show you that if you can get reproducibility in your manufacturing, you can hold live demos like I just did and got 0.29 milligrams per gram when I know the manufacturer thinks that they're shipping at 0.3 milligrams per gram. Well, 0.29 round it's 0.3 milligrams. The only reason I can get away with that is because of the reproducibility. And that's because, in fact, we put as much care into when we're manufacturing, we're really ensuring that we've got repeatability of that product. I think if you work with somebody like Zimmer and Peacock, you can get to market at a fraction of that 100 million that I've been suggesting. But realistically, if you're ever wondering what does it cost, how long does it take to get to market? At least 6 years. And what does it cost? If you do it alone, at least 100 million US dollars. So with that, I want to say thank you. I do appreciate many of you know this stuff already. I do recognize the names here, but I just wanted to, I appreciate you coming. I appreciate the support, and I will send out a recording shortly. Now, it is 7:45 here, so I have a couple of small boys to put to bed, so I have to apologize and end now. But if you've got any questions, when I send you that email later on, hopefully you can use my email address then. Okay, thanks very much. Take care. Bye-bye.